at University of Novi Sad, and then a PhD at University of Belgrade. And she's been working since 2008 at a institution called the Institute of Field and Vegetable Crops, which is very near Novi Sad. It's not part of the university, but a rather an equivalent to sort of USDA, government research institution. And she's visiting South Dakota State University on a roughly 9 or 10 month
Yes.
Postdoc, and she's working with Favina Matthew and other scientists up there. And her topic today is soybean diseases and the importance of certified seeds. And I'll just turn the lights down.
Yes.
Good.
Thank you, Mark. Hello, everyone. My name is Kristina Petrovic, and I'm the plant pathologist at the Department of Soybean at the Institute of Field and Vegetable Crops in Novi Sad from Serbia. But currently, you heard that I have a postdoctoral position at South Dakota State University in the lab of Dr. Favina Matthew. Today, I'm going to talk about economically important soybean diseases in Serbia in terms of climatic conditions and also importance of certified soybean seeds. So why certified soybean seeds is very important? You will see in this presentation that most of the soybean diseases also attack seeds and thus negatively affect germination and seed quality. Since everything begins from the seeds, it is necessary to use healthy seeds with high potential for a great yield. So Institute of Field and Vegetable Crops is recognized the importance of seeds 8 years ago when the breeding of the economically important crops such as wheat, sunflower, and corn started. And the soybean breeding program started around 40 years ago. And interestingly, some of our breeders work here in Iowa State University to learn how to breed. Today, Institute of Field and Vegetable Crops is considered as advanced soybean research center because it is a leader of soybean production where the best soybean genotypes in Europe are created. Our brand NSM has made that Serbia becomes recognizable around the world, and this handmade maps shows where our soybean seeds growing. More about the Institute of Field and Vegetable Crops you will see in the short movie.
Make the volume a little bit louder.
more interest in Serbia, it's just one of the prestigious prizes that the Institute has received.
Echo, did you like it? And since soybean crop is very popular, the soybean production area are rapidly increasing and therefore diseases are spreading and adopting the new conditions of the environment. Numerous phytopathogenic microorganisms parasitize soybean, causing various pathological changes. Their activity negatively affect quality, quantity, and yield stability, and in epiphytotic attack can jeopardize the profitability of growing of this industrial crop. More than 200 phytopathogenic microorganisms has been described with soybean, but it is believed that only 35 species can cause economically important damage. The most harmful are mycosis, followed by bacteria and viruses. It should be emphasized that Serbia and generally Europe doesn't have the big problem with soybean diseases, probably because we still haven't detected the prevalent pathogen that in some parts of the world, for instance, United States, can cause economically important damage, and a good example is Phytophthora soya. To avoid this situation, it is necessary to constantly monitor the occurrence and intensity of the disease attack, analyze the climatic conditions that favorable to development of diseases, also to create and cultivate the resistant and less susceptible soybean genotypes, and the most important, to use healthy certified soybean seeds. Since application of the fungicide in soybean in Serbia is not economically viable, it is necessary to respect all our technical measures. Through long-term monitoring of occurrence and disease intensity, today based on climatic factors, we can assume which pathogen will be present in the given year. So if you have a dry year, then the high intensity of checker rot can be occurred because this disease prefers dry and hot summers, while in the years with abundant precipitation, you can expect the increase of all leaf pathogens, then stem canker or white mold. And if you have the rainfall during the period of soybean harvest, then you can expect the increased percentage of fruit and seeds. In the year with heavy rain and moderate temperature between 64 and 71 Fahrenheit, the first symptoms which can be seen in the field is bacterial blight caused by Pseudomonas syringae glycinella. This disease is characterized by the yellow spot with a tan center, appears on the lower seeds, and over time these spots are large, emerge, and the whole leaf prematurely drop. Since bacterial blight causes the reduction in leaf areas, the losses can be up to 50% if in Serbia we identify also only raised 4. And in the breeding program of the Institute of Field and Vegetable Crops, it was included several lines with satisfactory level of resistance to this pathogen. Very often downy mildew is presented together with bacterial blight, and this is definitely the most widespread leaf diseases caused by host-specific obligatory pathogen, Peronospora majolica. This disease is characterized by the light yellow spot on the upper leaf surface, while under on the lower leaf surface, under the spots, form gray cover formed by oospores that constantly perform secondary infection in the field. The pods may be infected without any symptoms, and the seeds partly or completely can be encrusted with mycelium and oospores, and because that infected seeds is a major source of anoculum. The high variation in pathogenicity, it was observed within this pathogen. 40 different physiological races were identified, and this number is constantly increasing. And the Institute of Field and Vegetable Crops has 36 lines with a high level of resistance to this pathogen. The next leaf pathogen occurs later in vegetation is purple blight caused by Cercospora kikuchi. This disease is characterized by the protopigment, the completely colored leaf and soybean seeds. In 2016 in Serbia we occurred the more intensive attack of this leaf pathogen. So we don't have the reliable data of its direct impact on soybean yield, but according to literature, the losses are minor. This pathogen can negatively affect soybean seeds. Certainly, the most efficient and environmentally friendly measure for control all of these leaf pathogens is creation and cultivation of resistant and less susceptible soybean genotypes. On the other hand, you can notice in this picture that disease is showing also on seeds. So this is why we have to use certified soybean seeds. Otherwise, if we planted infected seeds, there will be a systemic infection of the plants that are completely lost in terms of yield. Now I'm going to talk about rot diseases, and economically most important is white mold caused by Sclerotinia spiraciorum and charcoal rot caused by Macrophomina fazolina. These two diseases is opposite, and that means that white mold prefers wet years, while the charcoal rot prefers dry years. So white mold is the most dangerous soybean disease because it causes premature wilting plants in the middle of the vegetation period, and the damage can be higher if the infection occurs during the flowering stage, when we can occur more than 50% of diseased plants, and this significantly reduce soybean yield. The disease can be occur in all growth stage of soybean, and this is a typical disease of soybean stem, where the light brown wet spot appear on the stem and spread, and the disease's tissue become soft, watery, and gradually eroded. Due to the interruption of the vascular plant tissue, the plant died, but it can be easily spotted in the field, where they found in the group covered with white mycelium, and the black sclerotia form inside of the stem, and sometimes also outside, by which pathogen survive for many years in the field. And this is the reason why we should avoid cultivate soybean in the same plot for 4 to 6 years, and especially after some other susceptible crops such as sunflower, because this is the common disease of these two crops. So this is the disease cycle of Sclerotinia, and you can see if we have the Sclerotia in the field, they will get apothecia with spores, with acid ascospores, and they perform the primary infection during flowering stage, and then another Sclerotia again form. Since we don't have resistant soybean genotypes to white mold, it is necessary to respect all agrotechnical measures, especially optimal sowing date and crop density, because a densely planted crop is prone to lodging, and that favors the infection. The opposite of Sclerotinia is Macrophomina fazolina, the causal agent of charcoal rot. And this disease prefers dry and warm summers. And thus, when temperature increasing, the infection and rapid spread of charcoal rot can be occurred. This disease can appear in all growing stage of soybean, and sometimes can colonize more than 50%, and that reduces soybean yield to 25%. This is a typical disease of soybean root, and the charcoal lesion can be occur on the lower part of the stem, where the small sclerotia, known as microsclerotia, form, by which pathogen can survive for many years in the field. So here we have the same rule like for Sclerotinia, and that means that we should respect the crop rotation, and also soybean should not be cultivated after other susceptible crops, such as sunflower, because this is also the common disease of these two crops. The most efficient measures for suppression of Macrophomina fazolina is irrigation, but with this we should be very careful, because irrigation accelerates the other soybean diseases, and especially white mold. Specific resistance to Macrophomina fazolina has not been identified, but it is known that some soybean genotypes possess moderate resistance or tolerance, And very often, drought tolerance mechanism is equated with chaparral rot resistance, and because of that, they recommended to avoid cultivation of soybean genotypes with late reproductive stage that fit with drought period. In addition to white mold and the chaparral rot, in some years, on the root and the lower part of the stem, we occur Rhizotonia and Fusarium root rot. It's very similar with the white mold and the chaparral rot because Rhizotonia prefers wet years and Fusarium prefers dry years. Among Fusarium species, the economically most important is Fusarium virguliforme, the famous blue isolate. And this disease caused significant damage here in the United States. And it was identified that this disease has a helper, and this is a cyst nematode, Heterodera glycinus. Also, it is commonly known that Fusarium species are facultative parasitized, and that means that they attack the plants exposed to stressful conditions such as drought. Fortunately, in Serbia, we don't have the cyst nematode, and Fusariosis appears sporadically, so the Fusarium species doesn't classify as some economically important species. However, I identified 13 different Fusarium species from root stem and soybean seeds in Serbia. We didn't find Fusarium virguliforme, but we found that 7 species showed a high aggressiveness to soybean seeds. And the last disease is the economically important complex of diseases caused by the Aporte species that prefers rainy years. And this is my favorite part because my research in Serbia and also here in the United States focused on the Aporte species. This genus is characterized by characteristic symptoms such as stem canker caused by the Aporte caulivara and the Aporte scalatis, then pod and stem blight caused by the Aporte soya and the Aporte longicola, and seed decay caused by all of these the Aporte species, and also other the Aporte species can be included. But the most dominant and the most aggressive pathogen of soybean seeds is the Aporte longicola. All of these diseases, the economically most important is stem canker because it is causing prematurely wilting plants. So this stem canker was identified in the areas located in the northern hemisphere. This is the reason why we call them northern stem canker. The first symptom can be occur at the end of July, and this is the time when pods creation of pods and grain filling. Near nodes, you can notice the brown spots that over time spread up and down along the stem 50%. In Serbia, we have a very strong attack 2016 because in that year the conditions was very favorable for development of this disease. In order to control, it is necessary to respect all our technical measures to create and cultivate the resistant and less susceptible soybean genotypes, crop rotation, and also using healthy certified soybean seeds. Unfortunately, there are no resistant soybean genotypes to northern stem canker, but we know that early genotypes are less susceptible, but not because of some genetic resistance, but because they avoid the moment of infection. So it is recommended to use that genotypes in endangered areas. Also, it should be emphasized that in addition for northern stem canker, we also have the southern stem canker, which cause very significant damage in the areas located in the southern hemisphere, and also we identified these species in the southern states of US. These two species phylogenetically is very close but geographically isolated. Considering that the climate change takes place, and in Serbia we very often have the very warm summers with temperatures above 100, we expect the occurrence also of southern stem canker in the near future. Regarding resistance, the situation is better because it was identified 4 resistant genes in different soybean genotypes. This is the cycle of stem canker. As you can see, the pathogens survive on plant debris where form Perithecea. They give the Ascendascospores, and they perform primary infection in the spring, very early in the vegetation stage, but you will see the first symptom in the middle of the summer, and after that the infected plants very fast become completely wilted and the new Perithecea again form. The difference between northern and southern stem canker is that southern stem canker firstly form Pycnidia with alpha conidia and then Perithecea, while the northern stem canker never form Pycnidia in the field. However, in North Dakota, it was reported that from the plants infected with northern stem canker, in addition to the Dioporte caulivora, they also isolated Dioporte gullier, known as sunflower pathogen. Also, in my study here, one isolate sample from soybean stem from New York State, tentatively identified as Dioporte ares. The next symptom is pod and stem blight caused by Dioporte soya and Dioporte longicola. So it is not possible to recognize which of them cause these symptoms in the field, and it's necessary to isolate them on PDA. The difference in appearance is obvious, and also Dioporte soya has a very similar disease cycle as stem canker. So the primary infection made by ascospores, while Dioporte longicola doesn't have sexual stage. However, Dioporte longicola sometimes produce very characteristic symptoms on the lower part of the stem near root, and that symptom we call black zone lines. Interestingly, Pycnidia form in this stromatic structure always relies both alpha and beta conidia, but if you make the isolation of the PDA, you will find only Pycnidia with alpha conidia. And it's very important to not confuse the black zone line with anthracnose caused by Colatotrichum species. The main difference is that Dioporte produce Pycnidia and Colatotrichum species produce acerola. And the last symptoms is seed decay. So the soybean seed decay infected with Dioporte species are very often very small, shrunken, flattened, elongated with cracked seed coat, and very often appear chocolate white of mycelium. The endosperm is also infused with mycelium, and the seed completely rooting. Sometimes on the seed coat you can find a stromatic structure with reproductive organs. Seed decay is mainly caused by Dioporte longicola, along with Dioporte soya, Dioporte caulivora, and Dioporte aspalati. But in Serbia, I identified for the first time some other Dioporte species, and this is Dioporte ares species complex, Dioporte noven, Dioporte peniculina, and Dioporte rudis. The pathogenicity trials show that well-known soybean pathogens such as Dioporte aspalati, Dioporte caulivora, and Dioporte longicola, as well as Dioporte noven, show the high aggressiveness on plant and also on soybean seeds, while Dioporte soya and Dioporte ares show moderate aggressiveness on plants but high aggressiveness on soybean seeds. Then the soybean seed is a source of different Dioporte species, also confirmed here in the United States. So here we identify well-known soybean pathogens, Dioporte longicola, Dioporte caulivora, and Dioporte aspalati. So we only didn't identify Dioporte soya for now, and I say for now because this project is in progress. But we found the new species, tentatively identified Dioporte congi, Dioporte eucrea, and one non-identified Dioporte species. Also, we performed the pathogenicity test, and this is the results after 7 days. The blue color showed the cases, the green color is the ungerminated disease, and the light green is germinal disease. So as you can see that Dioporte longicola, Dioporte caulivora, and Dioporte aspalati cause seed decay with more than 80% of the case seeds, while the Dioporte eucrea and Dioporte congi cause seed decay with 70 and 65%. Moreover, the isolate, the non-identified Dioporte species, didn't cause the decay, but if you compare it to control, it's obvious that the number of ungerminated disease is significantly higher. So we conclude that well-known soybean pathogens are associated with other reported species and they together cause seed decay. So as you could see, most of the soybean diseases also attack seeds and transmitted by them. So this is why we have to use certified soybean seeds. Moreover, Serbia and generally Europe is increasingly turning to organic production, and that includes organic breeding. So this is one more reason why we should keep the healthy soybean seeds and constantly work on finding resistance to diseases. With this, I want to finish my presentation, and also I want to take this opportunity to invite you to Serbia because 2020 is very important and very big year for us. When Institute of Field and Vegetable Crops organized 2 big world conferences, and this is World Soybean Research Conference and Conference of the International Sunflower Association. So I prepared a short movie about Serbia to show you why the world chosen us. lovely to enjoy.
Questions for Dr. Petrovich?
Fabina collected so many soybean stems from all different states last year, so you were involved to isolate those. My next question is, if yes, did you still see that northern stain canker just limited to the northern states and southern stain canker limited to southern, or they overlap?
Yes. We collected the samples from 21 states here, and we found that the northern part of the United States is northern stain canker, and only on the southern states we found this. In some states, this is the first report, but that will be published. And regarding the new, the approach species, they are only found on soybean seeds. And only the Porte Ares, this is only one sample from New York State that they detected from also from STEM. And we know that that sample is infected with northern stain canker, but we also isolated the Porte Ares. So we are working on that.
I'm curious about all of the species. Is your assessment of the species because we have better tools for separating them out, or that we can identify them now? And do you
Yes.
find multiple species in the same field sometimes, or?
I'm the part of old school, so
Yeah.
I prefer to see symptom in the field than I go into the lab, and the molecular tools show this. So from my experience in the past, we everything put in the Diaportosoia,
Yeah.
because someone wrote that Diaportosoia is heterothallic species, and it's not true.
Right.
So Diaportosoia is homothallic species, and everything what is different, we say this is variability inside of that species, but it's not variability. This represents completely other species. So molecular tools help us to resolve which species included there. Yeah.
Just to follow up to Charlie's question, are there like morphological traits that you've associated with these phylogenetic differences? So when you define a new species like Ares, is it just on the basis of genetics, or is there a morphological side to that separation of the new species?
Yes, morphological side is that I mentioned that the Diaportosoia is homothallic, so produce pycnidia with alpha and beta conidine, and after that Perithecea. But for example, the Porte Ares, this is the heterothallic species, the Porte Ares. So when I made the single conidial isolate, they produce only pycnidia, never Perithecea. So this is the morphological difference. But we found also that some other species that I can't say that they have fundamentally differences in morphology. So they are very similar, but it is commonly known that the Diaportis species is very hard to identify using morphological characteristics, because sometimes happen that today you perform morphological characterization one isolate, for about 1 week after you will have the different results, because the alpha colony is also changing. I see in the same isolate, I have alpha colony with one size, after 1 week is a different size. So they are changing also in which conditions you're keeping them, so it's very confused. But we are trying, I'm trying, for example, that I'm keeping everything in the same conditions to see. And the most important thing that the aborted species very fast become sterile. So they lose very fast ability to make some reproductive organs, and it's not possible to identify. So the CBS collection, for example, in Netherlands, has a lot of deported species, but they put only the number because they collect most of that.
So for the organic soybean production, besides the clean seeds, what other strategy you use? What other IPM strategy?
Yeah, we are starting to work on that because the big European project started on the 1st May or 1st July, and I'm the coordinator of that program inside of European project EcoBreed. So So want to turn to organic production that include organic breeding, and probably we will work on cover crops and no-till. This is the other technical measures, and probably include trap crops for insects, and so the natural things.
Do you also have an incidence of bacterial pustule disease in the soybean?
Sorry, I didn't.
Bacterial pustule disease.
Bacterial
of
pustule.
soybean.
Bacterial?
Bacterial pustule disease of soybean.
That we doing like that. We have the girl who is special bacteriologist.
Okay.
So I prefer fungi. I'm not familiar with bacteria, but the woman who works exactly on bacterial diseases, and also she performed study including the soybean, the bacterial bottom soybean. So she worked on that. I really don't know about bacteria.
Okay, I was just kind of curious because you only reported bacterial blight of soybean.
Yes, because I have my collection of resistant genotypes.
Okay.
Yeah. In breeding program in Institute, we include several genotypes with satisfactory level of resistance. So today we have 7 lines with a good resistance, and I'm charged for that collection. But I don't work with bacteria in the lab, so there's someone else charged for that. And she identified that we only have the race 4. Because you probably know that in the soybean we found the genes for bacterial blight, and you also have the 7 or 8 virulent genes that fit. But they only didn't found for race 4. So it's a problem in the breeding process.
Okay.
Another question. Can I ask a question about, you talked about, I think you briefly said that people will grow soybeans after soybeans for a number of years, or at least that happens sometimes.
Sometimes happen in monoculture, yeah.
Yeah. And so is there no general sort of rotation of soybeans with non-host crops? I mean, things that wouldn't be host to the same diseases like corn, that's not common?
Yes, but I think that in the rotation, but I don't know why people, something the farmers don't want to listen. You know, we're talking about that, we make extensions, we are going there, we talk about what they should do, but they doing how they want. So we have the problem, you know, when they put the soybean for 2-3 years in the same plot and then call us, oh my God, I have a problem, really?
Other questions to Dr. Cook? Thank you.